Hi, this is Miss Litton, and this is the final review for Unit 3. And we have done earlier reviews, and we've done all the essays except for 2 questions. So this first question we did not review earlier. We couldn't do this until we had talked yesterday, which reminds me I need to still put the notes in there for you. I'm sorry, I apologize. Remind me at the end of this. Describe the steps of protein synthesis beginning with transcription and ending with the release of polypeptide from the ribosome. Include in your answer a discussion of how the different types of RNA function in this process. So why don't we start with just the different kinds of RNA. How many different kinds of RNA are there? 1 second. I see a stranger. Okay, so the 3 kinds of RNA, what are they? tRNA, mRNA, rRNA. Good. All right. So the first one you said was T. What does the T stand for? Yeah, transfer RNA. So tRNA, okay, is transfer RNA. What does it transfer? An amino acid. So I'm just putting that there as an amino acid. It could, and it comes down, and at the base of it, do you know what that's called right there? Anticodon. Now, what is the codon complementary to? What is the anticodon complementary to? The codon. And what is the codon located on? On the mRNA. Now, they are not in groups of 3 like this on the mRNA. It's just continuous. I'm just doing that to simplify it so you understand it. So every 3 bases on here, okay, is called a codon. And this is mRNA. And what does mRNA stand for? What does that stand for? Messenger. Where does it get the message from? From the DNA. Okay? So this is messenger RNA. Now, if our 3 bases right here are C and G and U, here on the mRNA, what would be the bases on the anticodon? If this is C, the anticodon would have what? G. And if this is G, this would have C. And if this were U, this would have? Remember, these are RNA, so there is no T, no thymine. Instead, you have uracil. So you can think of this U as like a T. So who would T be bound to? A. Perfect. Okay, now this process where you're landing, let's just put, just for simplification here, let's say this is CCC. Then if we had another tRNA land here with its amino acid, what would the anticodon have to be here? GGG. Are we good? Okay. Now this whole process where tRNAs land on mRNAs and you start to hook these amino acids together to build a what? Protein. It's going to happen on the workbench. What's the workbench for this entire process? Yeah, so right here, you would have a ribosome. And a ribosome is composed of 2 things. What 2 things make up a ribosome? rRNA plus what? Proteins will build ribosomes. And ribosomes really are 2 units. There's a small ribosomal subunit and a large ribosomal subunit. Okay, so the 3 kinds of RNA, they serve 3 different purposes. 1, rRNA with proteins build a ribosome, and a ribosome is the site of the reactions for protein synthesis. mRNA, M stands for message. That's how you get the message out from the nucleus where the DNA is. Every 3 bases on the mRNA is called a what? Codon. And the codon is complementary to the anticodon located on the tRNA. T stands for? And it's going to transfer those amino acids to the message. And it's going to, the right tRNA has to land in the right spot because they're complementary to each other. And so the DNA dictates the mRNA dictates the tRNA, and the tRNA dictates the amino acid. We could look up those amino acids, what they are. What would we look up in order to find out what those amino acids are? Do we look up rRNA? Do we look up mRNA? Do we look up tRNA? What do we look up? mRNA. You know how I remember that? I'm looking up the message. So the thing I would need to look up is I would need to look up CGU on that table, and then I could find out what amino acid this is. Then I would look up C, 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 and I could find out what amino acid the tRNA has. Okay, now, ask your bio buddy who you're sitting next to. Anything in the Shadowlands on this part? Anything you need some clarification for? All right, so when we look at describe the steps of protein synthesis, beginning with transcription, what is transcription? The process of making what? mRNA, and actually any kind of RNA using DNA. But in the case of protein synthesis, I'm having you focus on making of mRNA. But the information to make rRNA and tRNA also comes from the DNA itself. All RNA messages come from DNA. Some of it will be expressed as a gene, and that will be the mRNA. So starting with transcription, that means you have to start with the DNA and the process of making mRNA. So let's think through the steps of making mRNA from the DNA. What's the first thing that's going to happen? Unwind and unzip. What can we use for that? Helicase, good. Now, I have two strands of DNA. I have a strand and a strand. What are the two strands I have? Leading and lagging. What one am I going to not even be concerned about? Yeah, I'll put that lagging strand away and I'll just use my leading strand. And instead of using DNA polymerase, now I'm going to use RNA polymerase. Now, it's got to know where to hook on. The place where it binds onto the RNA is called the what? Starts with a P. RNA polymerase binds to the promoter. Thinking it's promoting the process of building a protein. It's a promoter. Like, hey, come over here. All right. Okay, so RNA polymerase binds to the promoter. And then it goes along just like DNA polymerase did, putting the complementary nucleotide. But the complementary nucleotide it's attaching is an RNA nucleotide with a different sugar and one different base. Yes? And it's single-stranded. That's why I only need one strand. So when we said boom, 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 it's because I'm making that mRNA off of that DNA. Once I've got everything I need, now I need to pull this mRNA off the DNA and allow the DNA to zip back up, and I put it away. This mRNA that I just built is not good to go yet. I need to process it. What are the kinds of processing that happens to mRNA before it leaves the nucleus? What do you got to do to it? Yeah. Splice out the intron. I have to cut out whatever I don't want because I'm trying to manufacture a certain protein and I may not need the whole sequence. I might have started out in the nucleus with a much longer strand of RNA. And then what happens is you end up splicing a section of it out, and the part that I remove is the intron, and the part that remain, this could be exon 1, I don't know what I'm drawing there, and this is exon 2. I cut out the intron. What else do I do to modify this mRNA? I put on a what? Okay, a tail. Oh, sorry, here, I'll do it in a different color. A poly-A tail. And what else do I put on? A cap. And that cap is 7-methylguanosine. That's a cap. This cap will protect this mRNA. Okay, so I put on a cap, I put on a tail, and I cut out the introns before it leaves the nucleus. Leaves me with the exon. Allows me to take whatever DNA this came from. If this came from this DNA coding for this mRNA, the way I've spliced it now, I've paid attention to this part of the DNA and to this part of the DNA. But if I spliced it differently, if I took out another section over here, then I could have exon 1, exon 2, and exon 3, right? So I could use different parts of this DNA depending on how I manipulate it, on how I cut it. Okay, so now I make my mRNA. And now my mRNA can leave the nucleus, and it has to go out and hook up with the workbench. There's two parts to that workbench, a small and a large. Which one will it hook up to first? Small. Yeah. It'll hook up with the small ribosomal unit first, and then the large ribosomal subunit. And this process where the first tRNA lands, the second tRNA lands, and he kicks his amino acid off to the new guy, and then he leaves. And the way this works, it's the ribosome that shifts. Not the mRNA, but the ribosome that shifts. And so once this amino acid has been dropped off over here, and he's on this guy, then this tRNA, he leaves. And then we're going to read the next step, and another tRNA is going to land, and the ribosome is going to shift and shift and shift as it reads along the mRNA, building that protein until it reaches a what codon? Stop codon. And there are 3 stop codons that it can reach, and then you finish and you built your protein. Now your protein's not finished yet. It has to be folded right. It might even have pieces of it cut out, and we're going to talk about that in chapter 13. So now let's reread. Describe the steps of protein synthesis beginning with transcription. Transcription was this part, right? Right, that's transcription, making the mRNA and talking about how it's modified. Translation is this part, right? Building of the protein. You're translating it from a nucleic acid language into a what? Protein language. Okay, so we can talk about the steps of transcription, we can talk about translation, and I would probably start off my entire discussion just talking about the 3 different kinds of RNA. Yes?
When exactly does the mRNA become the tRNA?
Okay, the mRNA doesn't become the tRNA. If this were my DNA right here, my original DNA strand, there could be a portion on there, there will be a portion on there that's going to code for tRNA. A different portion on there is going to code for mRNA. And another portion of it is going to code for rRNA. And remember when we learned about the nucleus and the what's inside the nucleus? Nucleolus. What gets made in the nucleolus? rRNA. Because you need a lot of what? What do rRNA build? Ribosomes. You need a lot of ribosomes. Why do you need a lot of ribosomes? It's the workbench for building all your proteins. So that's why you make a lot of rRNA out there, is because they're going to come together to build your ribosome, which is your workbench. So in your DNA, you have areas. So this right here, where you have the mRNA, this DNA right here would be referred to as a coding sequence because it codes for a protein. These 2, where you build rRNA and tRNA, would be called a non-coding sequence because it doesn't actually code for a protein. It just codes for a worker in the process of building a protein. So it's called a non-coding sequence. It's not actually getting translated into a protein. These, see if you can get your brain around this. The RNA, the mRNA, and the tRNA are all transcribed, but only the what is translated. Only the mRNA is translated. The tRNA, rRNA, and mRNA are all transcribed, but only the mRNA is translated into a protein. Grasping? Questions? Ask it. If you have a question, probably somebody else does. Chances are. Ask me. Do you want to ask me? No? Okay. Any more questions on this one? Okay. So let's look at your next questions. Genes are located on chromosomes and are the basic unit of heredity that is passed from parent to child through generations. Can we accept that fact? Okay. So for each of the following mutations, describe how the genetic information is altered and predict one specific effect that could occur with that error. Okay, so let's think about the first error, nondisjunction. What's failing if you're doing nondisjunction? Yeah, one of the anaphases, right? Anaphase 1, where you're separating homologous pairs, or anaphase 2. So what's going to happen in the resulting cells? Some of those cells are going to have too many, and they're referred to as a trisomy. Some will have too few, and they will be a monosome. Turner syndrome would be an example of a monosome, because you only have a single X. Down syndrome would be an example of a trisomy. Okay? And the resulting effects, we can talk about what happens in each of those conditions. Okay, let's look at the next one. Base pair deletion resulting in a frame shift within an intron. Okay, so let's talk. An intron is an intron expressed No. Okay, so exons are expressed, introns are not. But now, think about this. This is base pair deletion. This is within the DNA. Okay, within the DNA itself. So what happens, that DNA has a mistake in it right there. Okay? But if the DNA has a mistake in it, who else is going to have a mistake in it? Right there. The RNA is going to have a mistake. And even if, I don't even know what I wrote there. Maybe. Okay? If I'm thrown off, even though it happens in an intron, basically what this is saying is a base pair deletion resulting in a frame shift. So that means that I am out one single nucleotide. But that means here, even though it was an intron part that will ultimately get removed, it means it's short one. So then, if that is in a codon, right? Codon is every 3 bases. Let's write it like this. What did I just write? 3-letter words. What is that representing? Codons, right? So if in the middle of it, even if it's an intron that you're going to take out, if I take off a letter right here, right? Now this first codon's fine, right? But instead of reading the big fat cat ate rat, now it reads the igif attic adapter at. Do you see how it throws all of them off? That's a frame shift mutation. All of them are screwed up, even though it happened in an intron, because I'm removing that part, it's throwing all of them off. Okay? A base pair substitution resulting in a different amino acid. If you have the wrong amino acid, how many different amino acids are there? 20 different amino acids. Some of them are hydrophilic, some of them are hydrophobic. What do we know about protein structure? Primary is the sequence of amino acids. Secondary structure, it could either be a what or a what? Alpha helix or a beta headed sheet. The tertiary structure is the folding pattern imposed upon the protein due to the what group? R group. So now if you have a different amino acid and it causes you to fold differently, now all of a sudden your protein is not going to function in the same way. Take an example, an enzyme, right? Enzyme has a particular shape for its functionality, and what do you call that special place on the enzyme you need to be that shape? What do you call it? The active site. If you have a different amino acid in there and you end up changing the folding pattern of that protein and changing its active site, it will no longer be functional and it will not be able to do its job. The wrong amino acid is evidenced when you talk about sickle cell disease. One amino acid has been exchanged for another, and the hemoglobin doesn't fold correctly, so the blood cell doesn't fold, isn't made in the right shape, and so you have all the ensuing problems related to that. Okay, so that would be 3. Let's look at B. Explain why it is more common for human males to be colorblind than females. Why is it more common?
Because if the female's the carrier and she gives the X to the male, or the father gives the Y and it's a male, he's only going to get that bad X.
Okay, I'm looking for another one.
Because it's sex-linked, he only has one X to depend on. And
Yes.
he doesn't get to choose between mom and dad, it's just mom.
Yes. We have 2 X's every single time, right? So I have twice as many opportunities to get it right than a guy. If it's a recessive disease. If it's a dominant disease, no. Okay? But if it's a recessive disease, which all of the sex-linked diseases I'm going to give you are going to be what? Recessive. So if it's a sex-linked disease, I've got 2 chances, as does every female in here. A guy only has 1 chance. So it's more likely for a male to get colorblind than a female. Okay, did that match what you were saying?
Yeah.
Yeah, exactly. She just said it in a different way. Okay, so now that's B. Let's look at C. Proper gene dosages are critical to normal development and function. Discuss the mechanism that maintains proper gene dosages related to the X chromosome in females. You know the answer to that. What do we do?
Is it the bar body? So we put one.
Yes, we put one away as a bar body. And that way, we only have one that's functional at any one time that we're using to transcribe and translate into protein, right? So that's how we even it out. A guy has one X that's working to make its proteins. A girl has one X that's working to make its proteins. We don't know which X it's going to be, but we only have one at any one time that is functional. Yes?
In the case of a dominant sex-linked gene, would the sex-linked gene, and say the female was heterozygote?
She'd still get it.
The
Yep.
bar body would still be the recessive one?
No, you don't know which one the bar, that's the thing, females are mosaic. We don't know which one is gonna be active. What makes us okay usually is because it could alternate in any one cell. If it's something we need, we're still probably making it correctly at least in half of our cells.
So they switch off this
Yes.
dormant group?
Yeah, yeah, you don't know, every cell could be a random X is what makes it. Now, could there be some sort of, epistatic, something above, that controls that or makes it more likely? Sure. We just probably don't understand that right now. Question?
I got answered.
Okay.
This isn't really related to that, but like, how do we get epistasis?
Okay, so epistasis would be an issue where, for instance, though there is a whole review on this, just so you know, okay? So if you have coloring in bunnies, okay? So, okay, I know that doesn't look like a bunny. So let's say big B anything, the bunny will be blue, okay? And little b, the bunny will be black. Are we all right with that? Okay, but then there's another gene called the color gene. And if the color gene, you have to have big C something in order for these to be expressed. Because if it says big C something, that says you may color me. What will you color me? Well, it depends on my genotype. If I'm big B anything, I'll be blue. If I'm too little b, I'll be black. But if there is no color, if instead it's little c, then it's not telling you to lay down the pigment. I don't care what the pigment says. It could say blue, black, orange, green, purple. We're not laying down pigment because we have the recessive gene, the gene that controls the laying down of pigment. Yeah. Mm-hmm.
They're kind of like baldness and like your hair color.
Yeah, exactly. Doesn't matter if you're supposed to have brown hair or blonde hair. If you get the gene for baldness, you have no hair. Okay. All right. So that was the second one, and that was it. The other ones I have already done in the review. Do you have any other clarifying questions you want to ask me that you're just like, Miss Lynn, I don't understand this. I want to know about this. This means you're 100%. You understand it. This is awesome. Can I put out a warning, please? This is something I have seen multiple times because this is your partner test. People lower what they normally do because they know they have a partner and they translate that into it will be easier. But the problem is if you both lower your game, you're both coming in a little less and you end up getting either the same score as you always get by yourself, or actually this phenomena happens. Do you know what it is? You get a lower grade. I have seen that so many times. Don't lower your game. Okay, study just as hard. What's today? Tuesday? So you have Tuesday, Wednesday, Thursday. Okay, so you still have 3 nights of studying that you can do. Raise your game and make this be a score where every mistake is caught and every essay is perfect. Okay, so you get a really good grade. Yes?
Was this an essay review part 2 or was this the test review?
I've been reviewing all along because there's a lot of content, so I would go back and look at those other reviews. And what I knew I was going to review today are the ones we couldn't review yet at the other essay review. So all the other essays are in there.
So there's not going to be a test review, like, it's just like...
Yeah, look at all of this. Yeah. Okay, have good days. Have a piece of toast if you need it. You're welcome, sweetheart.